Okay. Good morning, everyone. I am welcome you all to this seventh day of CSIR Life Science question and answer discussion session. Before I enter into the session, please let me know am I audible? Please type it in chat box. Am I audible? Can you see my slides? Is it visible? Good morning, all. So all those things are very important in this 5 cap of RNA. So 5 cap of RNA is require for the transport and the stability of RNA. And here the methylation process, we are all know methylation process happens, methylation process of RNA. For that we need a 3N. So we want to just understand basic concept about the that dash 5 dash ending, that all those process. Okay. And one more thing I will let you know in this RNA and DNA concept. First, we want to understand the what is the RNA function, what is the DNA function. That your CSIR CSIR related study. Please go through the all the things about the RNA and DNA functions, because that is the major and important basics in a molecular biology and fundamental process. Then next question, it's about the culture of an E. coli strain. The culture of an E. coli strain that is lysogenic. Okay, here the process called lysogenic for a phase lambda is grown at 32 degrees Celsius. This is the statement they provided. The E. coli strain that is lysogenic for Forge lambda is grown at 32 degrees Celsius. This is a statement. Induction of the prophage from the host chromosome will occur when the culture is exposed to. Okay, here the question, the statement they are provided, the E. coli culture lysogenic lambda fudge 32-degree. The question is whenever they are doing induction means stimulation or inducing process that called as induction of that is in the lysogenic. So they want to stimulate the prophage from the host chromosome. It will occur when it will occur. So that is a question. In the bacterial life cycle, that the prophage stage, that is it's not trigger at any stage that occur in one particular extreme condition itself. It will happen, not happen in the all the stages. Okay. So like here in a human, we are having the cancer. With cancer, we may get a cancer uh, at the one particular induction, right? Likewise, the bacteria is also having this induction, that expression, the induced expression with the specific condition. So they are provided, that is the lysogenic in phase, the is to index the prophage in the for a chromosome level. So if you want to make a change at a chromosome level, we want to use the ultraviolet radiation. So the answer is ultraviolet. Why we want to why I mentioned this basic stories means, at a 40 degrees Celsius, that is a heat, just it makes a change at the cellular level, not in a chromosomal level. It's here they mentioned they want to change the change at the chromosome level. If you want to attack at a chromosomal level, they want UV radiation, that ultraviolet radiation. And normal this prophase index, why we want to they are make a uh, recombinant, they can produce recombinant strains. For example, in this they like this question, they are producing any recombinant strain with this specific character. So for that they want to uv radiation, uv radiation for induction. Then next question is the that fidelity that the question is the fidelity of replicative base selection can be reduced by a factor of 10 power 2. Here this is the statement that that's fidelity of replicative base selection can be reduced by the factor of 10 power 2 when the repair of DNA synthesis involves. So the here the concept is the way they are the process is DNA replication process. In this DNA replication process, the DNA synthesis process, this DNA synthesis process get repair. So they may have there is some mistake, so they are doing repair. So during the repair um during this repair there is fidelity of replicative. So base replicative place means fidelity means here that is the assurance, assurance like yeah sure it is happen, like meaning that meaning is a fidelity of replicative, the confirmation replicative base after the repair. After the repairing process, the fidelity of replicative base selection is that reduced. So there was some mistakes are there. This is involves which enzyme, which happen by the which enzyme. Here the question is the all those things are happen the enzymes. We can't go with we can't do the any repair with our hand with our any instrument. In a DNA based all kind of repairs are involved with the all enzymes. In a molecular level, cellular level, we can't make the changes at a chromosomal level, make the changes at a DNA level. But that are all not possible with the any instruments or any hands. All those DNA based modification, restrictions, ligations, inductions, all those things are happen with the enzymes, proteins, chemicals. Okay, so likewise here they are provided, they ask the question is during this DNA synthesis which factor means which enzymes is helpful, which are used in the DNA repair process, that enzyme is reduced the fidelity of replicative. So this enzyme answer is the they provided TLS DNA polymerase TLS DNA polymerase. This is the answer here, that TLS RNA polymerase. Here some of the other enzymes also here. This endonuclease, all those things they are involved in the various steps in the DNA synthesis, DNA replication, all those things are happen. For example, here you can see this ABC XC nuclease. This XC nuclease is a UV specific nuclease, so specifically specific for the UV specific endonuclease. So they are called that are called as the ABCXC nuclease. Here this is not XO, this is the XC nuclease. It's a UV specific endonuclease enzyme. This also is helpful for the or that some proteins are not degraded by the UV, so that is respond that process, that proteins are made by this enzymes that are protected and that activate. And another one thing here is the DNA phytolysis. This yeah, that DNA phytolysis the also involved in this or photolysis. This kind of enzymes are involved in the or in that cells want to be face some kind of lights. because so we are not, as you may know, humans are not uh, not able to face the UV lights, particular amount of UV lights. We are not able to face the particular amount of our X-rays. Okay, if some of them may know, if you are taken the photograph, your lifespan become reduced. That is the basic one thing, that basic thing, it's help you to understand that all those the that oldest dialogues are made with the scientific background. So some we are not able to make this. So this light, that DNA photolysase, it helps for our the repair process. So this that also make convert the energy of light near the UV and visible ray, that is also the UV visible rays, and it makes the chemical, it's make that forms the chemical energy, and we can able to stable at a particular stage. And then that called as the AP endonuclease. I this in the AP endonuclease, that endonuclease means all those things you are involved in the are this apart from the nucleus process, all those this involved in the external proteins in this chromosome, that functional process, this involved in the DNA base excisions repair pathway. This also AP means, here they mention the A purinic and A pyrimidinic. The AP means A purinic and A pyrimidinic. Pyrimidine, purine and pyrimidine, and here the short, this enzymes are involved in the DNA base excision repair, that repair pathway. So this changes are happen with this enzyme AP endonuclease. Here okay, the one more thing I want to know here, they mention the called DNA, that TLS DNA polymerase. Here the called the TLS is a very specific, and it's in later here, it past 5 to 10 years, this concept, because the basic molecular biology explain all about the concept called concept called the DNA polymerase. But some of the latest researchers that explain the concept called the DLS, because TLS DNA polymerase, the enzymes which helpful for our that in the repairing, specifically they are involved in the repairing. But the concept, the one thing keyword here is, this enzymes are, that's TLS specific DNA polymerase enzymes are they are using that specific the translations. Okay, in the specific translations. In clear, I want to explain means, it's they are making the incorporation of nucleotide across the DNA damage. They're making the incorporation. So there was some misselection happened, because the if you want their medical application changes at one particular place. That another one thing here, the TLS, it's if I want to mention specific means, that are the transliterations, that makes a changing the particular one, that particular nucleotides as a particular one place. So only they are added. The next question is about what is the minimum number of what is the minimum number of NTP required for the formation of sorry what is the minimum number of NTPs required for the formation of one peptide bond? Here that's very easiest and direct question and basic common questions. One peptide bond during the protein synthesis. one peptide bond. Okay, peptide bonds are made between the or they are formed between all those sequences. In this sequences we can see our okay where okay, three amino acids are there in the one particular protein. If they are, sorry, three nucleotides are helps to form the uh, one amino acids, we are all know that. So three amino acids are helpful to form, sorry, three nucleotides are helpful to form the one amino acids. So this amino acids are having the internally specific peptide bonds. This peptide bonds are fully relevant to our the protein function and protein structure. So peptide bonding, how much amount of peptide bonds present here, this is the question. Whenever we are facing these questions, we can yeah, we can easily answer with this concept. Here their as per their question, here one three amino acids, so there are that three amino acids are make producing the peptide bond, one peptide that forming the peptide bonds. So there are four nucleotides are needed. Why means in between three, so that one that three makes a one amino acid, another bond is connected with another amino acid, so that nucleotide also necessary thing. For just imagine there is a single line, there are four nucleotides are there, that four in between four nucleotide, three are one amino acid. Okay, that one amino acid is responsible for another amino acid. Sorry, one nucleotide is responsible for another another amino acid. So there is a one peptide bond between this amino acid. So there are four NTPs are required for the this peptide bond, one peptide bond formation. Okay, next question about this thing is a single strand nick in the that parental DNA helix. The thing here statement is one single strand nick in the parent ahead of a replication fork, just ahead of the replication fork, causes the replication fork to break the recovery from his Clementi requires. Here the statement the provided is the a single nick in the parental DNA helix that present just ahead to the replication fork, that helps to break the replication fork. Here the recovery from this calamity recurs that so avoiding this uh, recovery means they want to stop or they want to change the habit. So for that we need to change the this process. For the change what we want to do means homologous. We are whenever you are doing the homologous recombination, you can easily stop this process, that is called the stop the this breaking of replication fork. This breaking of replication fork is stopped by the concept called the homologous recombination. This whenever you are facing this homologous recombination, you can this homologous recombination means that happens with the same happens between the same strand. Okay, in this homologous recombination, normal stage of homologous recombination, we are doing the homologous that changes with DNA same kind same that is the homo means same, that is same number of genetic recombination. That recombination happen with the happen at that is based on the exchange like process. Sorry. Okay, Then ligase, DNA ligase is helpful for the DNA attached. Site specific recombination means that is not no need to change, no need to make a homologous between two strand. That is one particular nucleotide or one particular site alone changed. That is the site specific recombination. Okay, next there a question is the amanitin inhibits. Okay, it's one that protein. The protein want to the inhibition factor. In this inhibition factor, which this is one kind of fungal protein. In this fungal protein, which this alpha amanitin, which this chemicals that like chemical this substrate, its substrates are fungal compound, that helps to inhibits the compound called the only RNA polymerase 2, that polymerase 2, which this why we it's inhibiting the RNA polymerase 2, what is the function means here. The sum of so if they are facing the fungal infection, your skins are getting some infection, so that all that easily influencing are the immune response. Why it's inhibit that is active, sorry, it's influencing immune response means whenever the one protein and one chemical or one molecule inhibiting or stimulating a RNA polymerase, there is a misprotein function happen. The protein function is disturbed. Due to this disturbance of this protein function, there are some unwanted immune responses are generated in the human body. This make allergy reactions. So this fungal this amanitin, it's a chemical which derived from these are this fungal compounds, that is a mushroom like uh, organism, it's producing this common compound. Then reverse transcriptase have both ribonuclease and polymerase activity. It's a statement. Okay, that is the statement called the reverse transcriptase has the both ribonuclease and polymerase activity. So here they asked the question with a ribonuclease activity is required for what function? Why the ribonuclease is required? For the degradation of DNA strand. Why it's want to be degrade? After the protein synthesis, that want to be cut at one particular place, otherwise there is a continuous, no, there is, we can't produce the various proteins, that is become accumulated. So whenever they are producing one protein, after production of synthesis of one protein, that ribonuclease is active on the DNA and cut down at the particular strand. Okay, next question is about in the gene regulation open reading frame. We are know the open reading frame implies. What is the answer? This is a definition. The gene regulation open reading frame implies a series of triplet codon not interrupted by the stop codon. It's a open reading frame. It's a series of triplet codon not interrupted by the stop codon. This is the answer here. This is the definition for this open reading frames. Then while replicating DNA, the rate of misincorporation by DNA polymerase is 1 in 10 power 5 nucleotides. Here the question is while replicating DNA, the rate of operation by DNA polymerase is 1 in 10 power 5 nucleotides. However, the actual error rate and the replicators in one is 1 in 10 power 9 nucleotide incorporated. Here the two statements are there. While replicating the DNA, the miscorporation of DNA polymerase is 1 into 10 power 5. In the actual error recur means replicated DNA in a 1 into 10 power 5, 9, 9, 1 in 10 power 9 nucleotide in incorporated. This achieved mainly due to the statements is just complicated, but the answer is very easiest one. How we can understand the answer? Okay, what is the replication process? Replication is it starts from the 5 prime end to 3 prime end. So that is the some incorporations are happen by reincorporation happen by the DNA polymerase. Okay, so DNA polymerase activity is it makes some activity change on the sequence. Another type, they are doing the some of the error happen in this nucleotide sequence, and the replication process it's incorporated. So the statement is fully depends on what we want to understand. Whenever the DNA replication happens means, first what the DNA want, that molecule, that DNA converting, DNA replicating molecule want to do. So first we want to understand what is the that responsible molecule. So here the responsible molecule is a DNA polymerase. DNA polymerase just find out all the replication possible replicating sites. So it's want to be doing the 3 prime dash to 5 prime dash proofreading activity, that want to be do the proofreading means that want to synthesis, that want to convert all the nucleotides in and producing the alternative sequences. And another important thing, a question, a concept called as this amino acid selenocysteine is incorporated in peptide chain during the translation. This kind of studies are not it's not familiar, but that is a rare study. But many of the upcoming researchers are focusing on this concept is important. Whenever you are doing the translation, we want a protein, if you want any vaccine, if you want any antibody production, if you want any insulin production, there we are modifying the translation process. Whenever you are doing the translation process, that is the addition of amino acid selenocysteine. Uh, this process by it, how it is happen, this process happened by option D, the changing of serine into tRNA. This is the one, the changing of serine into tRNA serine, modification of serine into selenocysteine, and then incorporate through a specially placed stop codon. This is the overall rare one. As I am said previous day, one of the student asked the question, I would like to buy this book, shall I buy? Okay, that in that case, whenever you are studying the concept by in the book, come narrow down. You are just know the previous question, that 39th question, that is a open reading frame question, is open reading is a def, you all know, easily understand and know by the one book. But when this major questions are like come like this, they are asked based on the research, current research. So whenever you are studying the CSIR online CSIR subject, CSIR topics, just go through the whole book concept and then do the research with your with the concept called research study. Okay, if there is a topic called the gene function, gene protein activity, in that case you can go to the Google Scholar and type the particular concept. There are some research papers are there, review papers are there. Select the review papers. That review papers contains more more information. That review paper should be in past 5 year, or that paper may published in 2018, may published in the 2020, that is very important. So Whenever you are, if you want to study, understand the whole concept, just go to the Google Scholar and type it amino acid selenocysteine means you may get a research review paper. Don't to Google, go to the Google Scholar. In that you will get the UV paper and go through that review paper and get more knowledge about this basics of the molecular biology. The fundamental process is the basic. In this fundamental process is relevant to the fully relevant to the molecular biology. The studying of molecular biology basics is not enough for the CSIR examination. And the next question is about the site specific recombination results in the precise DNA rearrangement, which is limited to specific sequence. This is the site specific recombination results in the precise DNA rearrangement, which is limited to the specific sequence. The enzyme that or carry out the process are. So here the question is, they are provided the statement, ask the question. The statement is enzyme. Which enzyme is important for this DNA rearrangement? So, for DNA rearrangement means that important thing. Site, they are mentioned one more, the term site specific. For site specific recombination results, the DNA rearrangement. Sometimes the specific sequences are limited. That is because of the R nuclease and ligase enzyme. And which of the forms is not true about small interfering RNA? Okay, it's very interesting. That's the definition based question. Small interfering DNA, RNA. It's RNA. It's all about the siRNA except B option, that are odd definition of siRNA. siRNA has a 21 to 25 amino acids, nucleotides, 2 nucleotides overhanging the 3' end. Then another definition is siRNA is often induced by virus. siRNA does not generally act at a level of transcription. So the wrong answer is siRNA is processed by RNA DNA complex. Presence of an internal ribosome, and it's a research based question. Presence of internal ribosome NT site, that is the IRSC in mRNA. It promotes the translation under the adverse condition. Normal is a normal condition, it's an adverse condition. In this during this adverse condition, the presence of internal ribosome and NT site is helpful for the RNA to promote its translation. Okay, this is all some concepts about our CSIR, that unit third. Here, that is fundamental process. That fundamental process are related to amino acids, DNA, RNA, replication, translation, transcription, all those processes like molecular biology, system physiology of plant. This whole concept is fully related to the of plant. Okay, light is the dominant environment signal that controls stomatal movement in a leaf of well watered plant. Here the mentioned statement, leaf of well watered plant grown in a natural environment. Which one of the following wavelength of light is responsible for such a regulation? So here they provided the two statements. One is the light is a dominant environmental signal that controls the stomatal leaf movement, the movement of in the plant grown in a natural environment. Which of the following wavelength of the light is responsible for such a regulation? So here the answer is a blue light. Okay, the blue light is blue light means it comes like it's in a particular range called 300 to 400 nanometer. This 200 to 400 nanometer. This kind of blue lights are helpful for the their way that signals, environmental signals receiving. Which of the following is not main factor that contributes to water growth under normal condition? The question is main factor that is which is a not the main factor contributes the water potential during plant growth under normal condition. So whenever they are living in a one particular place, the plant growth under normal condition needs solute potential, hydrostatic pressure and gravity. Without the temperature we can't do any contributions. So the factor which not involved in this water potential means that is the temperature. Then another thing question is the plant hormone indole acetic acid is present in most of plant. The plant hormone indole acetic acid is present in the most plants. Okay, we all know this. The structure of the hormone is related to which one of the following amino acid? Structure of the hormone. Okay, indole acetic acid structure is for depends on the which amino acid, that is tryptophan. Okay, the indole acetic acid structure, just google it, indole acetic acid structure and compare with the tryptophan structure, that is related to the tryptophan structure. Sorry. Which one of the following is not characteristic property of carotenoids? Carotenoids, they are the phytochemicals which present in the plant. This carotenoids helpful for, they are integral constituents of thylakoid membrane. Carotenoids are integral components of thylakoid membranes that are necessary, that called as accessory pigment. Carrot having carotenoid, that is in that orange color, that having the carotenoid. So these are all the pigments. Then they are protect plant from damage caused by the light. So they are protecting the plant. So which is not character? This possess complex porphyrin ring. Okay, this is the not statement. So carotenoids don't having the complex porphyrin ring. The carotenoids don't having the uh, porphyrin rings. Then next concept in the plant physiology is which of the following statement about LEAFY? That is one statement. Here they are asking the that that regulatory gene of this is the statement of, this is a gene that called as the LEAFY. Shortly it's known as the LFY. This regulatory gene in Arabidopsis thaliana is following statement is correct. About this Arabidopsis thaliana, this one more thing, just before and like based on this Arabidopsis thaliana, like plant physiology unit, just to go through this Arabidopsis thaliana concept. This Arabidopsis thaliana concept, they are asking many questions in an exam. That is a basic thing. Arabidopsis that all those things researchers, genetic researchers are very important. So the regulatory gene in Arabidopsis thaliana, which is correct. Okay, they ask the correct question, is involves in the floral meristem identity. That this gene LFY is involved in a floral meristem identity. Okay, next question is the our concept called nitrogen gas is reduced to ammonia by nitrogen fixation method. This is the statement. Nitrogen gas is reduced to ammonia by nitrogen fixation statement. In order to execute the process, which one of the following compound is usually required? For the nitrogen fixation, we want to understand the pathways which involve in the nitrogen fixation. So nitrogen fixation pathway, there are ATPs are used in the nitrogen fixation pathway. This is the basic concept in this Nitrogen fixation process. Important C4 pathway, C3 pathway, C4 plant. What is C4 plant? What is C3 plant? And nitrogen fixation pathway. That all those things are very important. In this nitrogen fixation process, there are two ATP molecules are required. They are produced in this process. Okay, after this reduction. And next question is the quantum field of oxygen evaluation during photosynthesis drastically drop and far red light. Here the statement is quantum field of oxygen evaluation during photosynthesis. It's drastically drop and a red light, far red light. This far red light, they are met the reduction. So this reduction is this effect is known as they are called the red drop. Okay, red drop, that it's called as red drop. So the here they mentioned the far red light reduction, but that are called as the red drop. Don't that is a far red drop is not answer. Red drop is the answer. Is not a part of pyruvate dehydrogenase enzyme complex. The pyruvate dehydrogenase enzyme in a glycolysis pathway. As I mentioned, we want to know glycolysis pathway, Krebs cycle, all those things are very important. So just remember those concepts. So which enzyme is not a part of dehydrogenase enzymes complex in uh, glycolysis pathway? Not a part of. So here they are using pyruvate dehydrogenase, dihydrolipoly transferase, dihydrolipoly dehydrogenase, but they are not using oxidase. This dihydrolipoly oxidase. The dark grown seed. Next question is a dark grown seeding display triple response. They are showing the triple response when exposed to ethylene. Here the statement is whenever you are doing seeding, means dark grown seeding using the ethylene, and this ethylene when which one of the following state is a not part of triple response? Well, whenever you are seeing the question itself, you can get the answer. What is the triple response? First we want to know the triple response. Here except the answer B, all those things are about the triple response. Triple cortical elongation, that decreases the epicortical elongations, then thickening the shoot horizontal growth of epicortical. So it's which is not relevant to triple response, means that is the answer B, rapid unfolding and expansion of leaf. Rapid unfolding and expansion of leaf. And the next question, the chloroplast, that site of coupled oxygenation reduction reaction. So very easiest and interesting one. They are provided the clue, that is the chloroplast, the site of coupled oxidation reduction reaction is the which is the thylakoid membrane. The chloroplast are or it's this process, oxidation reduction reactions happen in the thylakoid membrane. And the next question is which one of the following statement regarding seed germination? It's about the seed germination of wild type plant. Wild type plant means that is that is the ancient one, that is the initial parental one. So which one of the following statement regarding seeds germination of white type plants is not correct? They are providing the not correct. Question asked like not correct. Here the answer is B. Which one is not correct? That seed germination is not light accomplished with high temperature can break the seed dormancy. It's a wrongest one. Okay, whenever your seed germination is a basic one, if you are doing that green nuts green pies grown in your home, that you are taken some amount of peanuts and pour it dip it in water and collect that and tie up with kerchief or any cloth. You are make an bundle and kept it at overnight means there is no light, so in that night getting grown and it's like that's it's a healthiest food. So these things are very nature and basic questions. So the here that it accomplished means there is a high light required, high temperature. With the high temperature, this is a wrong statement. When in a high temperature the seeds are not able to germinate. Okay, then what is the correct one? The low APA and high bioactive GA. These things are that they are like indolacetic acids. There are some hormones are there that are GA and APA can break the seed dormancy, and GA induces synthesis of hydrolytic enzymes, and another enzymes in the cereal grain. Another option is its degradation of carbohydrate and storage of protein provide the nourishments and energy to support of seeding growth. These things are about the seed germination. In the seed germinations we want to know there's some basic concepts about which enzymes are involved, which condition we can do the seed germination. All those things are very important in this seed germinations. Okay. Mhm. Oh, I actually there is a and hormones and functions. There is a one table is there. They are providing the hormones and give the answers. Just Google it. What are responsible for seed germination? There you can see easily. You can get better comparative results. Okay, all those the regulation of seed dormancy, they are this APA and the GA is involved. That APA means that is the ascorbic acid. This GA, that is the gallic acid cycles. The both things, gibberellic acid. GA means that is a gibberellic acid. APA means that is ascorbic acid. When this ascorbic acid is it's sometimes it inhibits the seed non-dominant. Okay, dormancies are the mature dormant seed seeds are they are produced. Sometimes some APA are not, so only they are inhibiting the seed germination, so they are in a low quantity. They want to be in a low quantity, so only the statement A they are mentioned that is should be in a low and higher G. The gibberellic acid is that we are need it in a higher amount. That is default. That is typical seed germinating seed that makes us seed germination. And the next question is the duration of operation of C2 oxidative photosynthetic cycle. Which of the following metabolites is transported from chloroplast to peroxisome? This is a very basic and a theory question. We want to we want to study it. During this C2 oxidative photosynthesis cycle, the glycolate, it's transport from cytoplasm to perisome. Then Arabidopsis thaliana seeds were planted on the uh, moringa Morangioscus plates. This is the MS plate that it's an experimental thing with and without hormone the medium. This is the statement. Seeds were found to germinate late. Okay, there is a delayed germination in a hormone containing MS plate. There is a hormone present plate, so it should be want to grow fast, but there is a delay. It's compared to the MS plate without hormone. Identify the hormone. If they are here, this as per the previous discussion, you can answer the hormone with and without. which hormone is delaying the seed germination? We all know ascorbic acid, APA, it's delaying the seed germination. It's a very basic concept, but they are provided with para like question. That is okay. This is a big question. I don't know the concept. You thought like that, but this question is very very easiest concept. The basic thing, which hormone is helpful for high seed germination? Gibberellic acid is helpful for the higher seed germination, and the ascorbic acid, it's lowering the seed germination, so only it get some delay. The hormone with plate, the plate with the hormone, it takes long time. Plates without hormone, it's without this hormone, it's they are not affected the seed germination. That is, seeds are normally germinated. And some more concepts here, that axine, cytokines are involved in the seed germination also. Ethylene signaling pathway is important in the fruit ripening. Which one of the following response is routinely used to identify ethylene signaling pathway? This ethylene concept is involved, that is important in the fruit ripening. That fruit ripening pathway is helpful for the triple response. In the previous okay, triple response, here you can see they mention this ethylene concept. So this dark signaling seeding display, this is the triple response process. Here they ask again the question. This is the another year question. In that itself they are asked the ethylene based question. This ethylene based question, just please refer whenever you are free. The triple response, so it's related to the ethylene pathway. Then which of the following mechanism is not involved in the providing photo protection to plants? The photolysis of water is not preventing the photo protection. It's not providing. Photolysis of water is not providing the uh, photo protection of plants. Okay, what is which will give the protection? Degradation of D1 protein, zeaxanthin formation, thermal dissipation. These are all the things are involved in the photo protection of plant. Under which condition of member of family Germinase synthesis, graminase synthase and release photosiderophores, this releasing the photosiderophores, which under which condition it can able to produce iron deficiency. Whenever the this family graminae family of plants are facing iron deficiency, that are producing photosiderophores. This photosiderophores having many medical applications. The phytochemical, that is the chemical compound which produced in many plants, that plant should be in a iron deficiency condition. So whenever the plant which under present under the iron deficiency content, that plant producing the photosiderophore, graminea plant can produce the photosiderophore. Those who, is any persons are in a research field, you can try this graminea plants are produce the photosiderophores. The membrane of a ceiling sensitive plant are characterized by the higher proportion of saturated fatty acid. If there is a higher proportion of saturated fatty acids present on the chilling sensitive plants, which of the following plant hormones can mimic the TET1 mutation, causing de-ethylation and chloroplast development in dark condition? The thing here is a dark condition. Whenever they are plant undergone a dark process, that is a Krebs cycle process, that process under the cytokine activity. Then which one of the following statement is incorrect about the role of oxidative pentose phosphate pathway? The thing here a statement is the oxidative pentose pathway of plant metabolism. Here the wrong answer is D. I am eliminating this. The which of the correct answers? Role of oxidative pentose pathway is generating NADPH. They are producing the NADPH required to derive photosynthesis reaction, production of pentose phosphate for the synthesis of nucleic acid, formation of erythrose 4 phosphate of for the synthesis of aromatic amino acids. So which is incorrect? It's producing NADH to generate ATP. This is a wrong thing. The NADH is not able to produce ATP. NADH, if the NADPH is converted to NADH, that is a phosphorylation process. ADP only produce ATP. It's the wrong thing. The production of NADH to generate ATP. Then duration of photosynthesis carbon reduction cycle is green leaf. The net production of one molecule of glyceraldehyde 3 phosphate requires one of the following combination of energy equivalent. So when the statement is bigger one, but the concept is carbon reduction cycle. In carbon reduction cycle, they are producing 6 NADPH and 9 ATP. The very easiest and simple thing. Please note it down. Whenever you are you do studying this cycles, just note it separate. Make a separate note. Okay, this carbon reduction cycles make a produce utilizing this amount of NADH and NADP, ADPs and PDPH and this amount of NADPs. Here they in this carbon reduction cycle leaf, they are producing the 6 NADPH and 6 9 ATPs. And which one of the following essential nutrient is associated with urease enzyme found in higher plant? The micronutrient is a nickel. The plant which utilizing nickel that associated with the urease enzyme containing plant. The plants are able to perceives light through the various photoreceptors and downstream the genes. Which of the following gene is not involved in a light perceptions? This gene related concept here, the gene related concept, the NPR1 gene is a photo. It's that are downstream the gene and it's receiving the precise light. Okay, this is not involved in the perception light perception. This NPR1 is not involved in the light perception. So which are all the involved in the light perception? PIF3 and FPI and CRY. These are all involved in the process called the light perception. The NPR1 is not involved in the light perception. Okay, thank you everyone. Today we are discussed unit 3 and 6. And welcome you all to this the tomorrow class. So please join with us on tomorrow. If you have any queries, type it in chat box. If you have any want any clarification, let me know through the telegram or mail. Thank you everyone. Thank you so much. Okay, this is ready. I will note and send the PPT and notes. No problem. I'm noted your mail ID. I'll send the mail. Okay, the session. So